Okay, thank you very much. Over the next 20 minutes, I will try to give you all relevant information in humans and animals where human data do not exist about a newly proven complication of full mechanical support of the ventilatory pump, which is called ventilator-induced diaphragmatic dysfunction. And we have, over the 10 years ago, nearly, we have defined that as the loss of diaphragmatic force generating capacity of the, that is specifically related to the use of controlled mechanical ventilation. Well, now we have human data showing that this is a real phenomenon in humans. If you take brain donors, brain dead organ donor patients, and you measure the tweets, pressure developed in the airway upon magnetic stimulation of the phrenic nerve, which is a good measure of transdiaphragmatic pressure development, especially in the absence of excess auto-PEEP, intrinsic PEEP. You see that over the days of mechanical ventilation, quite quickly they develop a decline in the force generating capacity. This has nothing to do with motivation. Patients don't really try it by themselves. It's just the contractility, objective contractility. And you see that there is a time-dependent decline in the contractility. Now, what is the mechanism of that contractility? And I was talking about that during the roundtable before. Is it an electrical problem? Is it a problem of the brain sending the message to the phrenic nerve of the diaphragm? We don't have the human data to say that, but in animal experiments, you see that when you stimulate the phrenic nerve and you are measuring the EMG of the diaphragm, this is 1 day of mechanical ventilation, full mechanical ventilation, 3 days, 5 days, you don't get any increase in the latency. So the transmission is okay. What you get is a reduction in the EMG amplitude, which says that the problem resides within the contractile machinery. So what could the mechanisms of this contractile dysfunction be? And despite a lot of research, the main three mechanisms are muscle atrophy, oxidative stress, and structural injury. Well, let's go now first to the muscle atrophy. This is the landmark paper by Sanford Levine in the New England Journal of Medicine. And you don't have, these are again brain dead patients that were organ donors and they received diaphragms. This is what they call case in that study. And here you have the cross-sectional area of the myofibers of the diaphragm, both slow and fast myosin, type 1 and type 2. And here's the control situation. And patients not being subjected to prolonged periods of full mechanical support of the diaphragm. And you don't have to be an expert to see that there is a big difference in the cross-sectional area. And not only is there a big difference in the cross-sectional area, here is a different set of patients. Again, the same human model, brain dead organ donors. Again, the same reduction in the fiber cross-sectional area. But the most important thing is that it looks like being a progressive phenomenon. The more you prolong controlled mechanical ventilation, the less the cross-sectional area becomes, the more the atrophy of the muscle of the diaphragm. Now, to have atrophy in the diaphragm, there is only two ways that could combine themselves. We could either have a reduce in the protein synthesis or an increase in the proteolysis. And in humans, we don't have any evidence that there is a reduction in the protein synthesis, but in animal models, that's clear. Here is the synthetic rate of myosin heavy chain, one of the most important contractile proteins in the diaphragm. And you see spontaneously breathing rats for 6 hours, 12 hours, and 18 hours, and respecting the corresponding animals that have been subjected to equal time periods in controlled mechanical ventilation. And you see that from the first 6 hours, you have a reduction in the synthetic rate of myosin, less proteinosynthesis, that is lasting as long as controlled mechanical ventilation is lasting. So it's not a transitional phenomenon. Now, do we have any evidence of increased proteolysis? We have very solid evidence of increased proteolysis, and in fact, eukaryotic cells have three main systems to make proteolysis to degrade proteins. They are the calpains, the proteasome, and the autophagy lysosomal protease. Well, do we have evidence of activation of the calpains? Clear evidence in humans. Here you have samples from diaphragm from humans subjected to controlled mechanical ventilation. You have 3 different isoforms of calpain that are existing in the human body, and you see a huge activation in the expression, a huge increase in the expression of 3 calpain isoforms, which suggest but are not proofs that calpain are active there. But if you measure the calpain-like activity of the 20S proteasome, you get a definite increase. So, at least in animal models, and in humans. So this means that the increased calpain expression is also leading to increased calpain activity. And calpain activity is very important because you cannot really degrade the contractile proteins in the muscle because they are very well organized into actin and myosin filaments bound to titin and other proteins. So first you have to partially degrade this contractile machinery to smaller pieces, and these are made amenable to the second major degradation system that we're having, which is the proteasome. Well, the proteasome in the human body exists in 2 different isoforms. It has a core cylindric structure that has 2 different proteins, A and B subunits, and it has 2 caps, 2 regulatory caps. This is 26 proteasome, this is 20S proteasome, and they're doing some like a different work. This is the normal proteasome working, and for that to degrade a protein, first the protein has to be targeted with ubiquitin molecules. So ubiquitin molecules are being added to that protein through a complex process of various ligases in an energy-dependent manner, and then this makes the protein unfold, enter the core of the proteasome, and become degraded into the amino acids. So here is how it happens. You have the ubiquitinated protein that is able to unfold and enter the core of the proteasome where the cleavage takes place. Now, do we have evidence for this activation of this system in humans? Yes, we do. This is from Levine's initial study. 2 different ligases, atrogene and MARF1, are highly upregulated at the mRNA level on the diaphragm. And similar results were obtained a few years later by our group with Sabah Hussein. Atrogene and MARF1 were highly upregulated and other components of the proteasome system. And in fact, it is a relatively preferential activation of the system in the diaphragm because if you take the quadriceps muscle, you have some atrogene but no MARF activation. So you have greater activation earlier on of the proteasome in the diaphragm. Now, we have upregulation of the expression of the ligases. Are they active? Well, at least the proteins that are ubiquitinated, so they have been made targets for proteasome degradation, are increased in the diaphragm of patients that have been subjected to controlled mechanical ventilation. Here is the blood, and here is the average. One study, second study, different group of investigators, exactly similar results, an increase in the total level of ubiquitinated proteins, proteins that are ready to be degraded by the proteasome. The third system that we are having to degrade proteins is the lysosomes. And lysosomes can degrade both invading proteins from outside of the cell, but when the cell is being stressed and it reaches a critical energetic condition, then it starts degrading itself in a process called autophagy just to provide adequate nutrients to survive. So autophagy could be a third mechanism which could lead to death and injury, which is different from apoptosis, which is programmed cell death, or necrosis, which is death upon exogenous severe insult. And we have provided evidence that the autophagy system is being upregulated after controlled mechanical ventilation in humans. The various ligases that I don't have time to go into detail, but similar to what is happening with the proteasome, the autophagy system works by a chain of reactions that in fact causes an initial phagopore to engulf the organelles that is going to degrade and create the autophagosome, and finally it fuses with the lysosome to make the lytic environment. And you have a definite upregulation in various enzymes that are implicated in the autophagy process in the diaphragm. And you have definite proof at the electronic microscopy level of autophagolysosomes formation within the diaphragm. And in fact, the longer the duration of mechanical ventilation, the more the upregulation we are getting in the level of autophagy-related genes, which says that autophagy is a progressive process upon controlled mechanical ventilation. So we come to the next issue, oxidative stress. Well, now we have clear evidence in humans that we have oxidative stress within the diaphragm, and we have difficulty measuring the oxidative stress, so we usually measure it in tissues by its footprints on proteins or lipids, by the footprints on protein oxidation, carbonylation, or lipid peroxidation. And here you have the corresponding blots of carbonylated proteins in the diaphragm. These are Western blots, or hydroxynonenal protein adducts, which is an index of lipid peroxidation. And you see that both the carbonylated and the lipid peroxidated proteins are highly upregulated in the diaphragm, subjected to controlled mechanical ventilation in humans. Why is that important? This is important for one very specific reason. Oxidized proteins default by themselves, unfold, and can enter the 20S proteasome and become degraded. So this makes them prone to the degradation at atrophy. But not only that, this is animal experiment, not a very direct proof, but very, very important. Here is the blot. You separate the proteins by their molecular weight with electrophoresis of the diaphragm. The sample comes from a rat. And then you probe with an antibody that reacts with reactive carbonyl derivatives. So footprints of oxidative stress. And you see a very strong signal around 40 kD and a very strong signal around 200 kD. Then you strip the membrane, you remove that antibody, and you reprobe for actin. And you see you have an upregulation, and the signal acts exactly the same molecular weight. Then you strip and you reprobe for myosin heavy chain. Again, great signal, which suggests that probably actin and myosin are targets of oxidative stress within the diaphragm of mechanically ventilated patients. And in fact, we don't have direct evidence that actin or myosin are oxidized, but they are definitely reduced in the diaphragm of patients subjected to controlled mechanical ventilation. Here you have the reduction in myosin heavy chain, the fast isoform, and the slow isoform, and actin. So all contractile proteins are reduced in the diaphragm of patients subjected to controlled mechanical ventilation, and probably oxidative stress definitely contributes to that process. Not only that, but we have change in the characteristics of the ultrastructure of the muscle that is keeping the muscle architecture in an appropriate environment. Here are results by Leo Hyung's group here from Nijmegen, where they subjected animals to either controlled mechanical ventilation or spontaneous breathing, and they were measuring the pulsive force. And they have seen that even the pulsive force during mechanical ventilation goes down. You have a depression in the pulsive force, which means that controlled mechanical ventilation reduces the stiffness of the diaphragm. And because the most important protein for the pulsive characteristics and the architecture of the muscle is titin, they thought that titin might be reduced, which is not really the case. But when they did the clever experiment, say, okay, now we're not going to measure the total expression, but the phosphorylation status, which is the most crucial factor for titin activity, they found that, in fact, if they give a phosphatase, they cannot really change the passive characteristics of force production in mechanically ventilated animals, whether they can really decrease these characteristics in control animals, showing that titin is dephosphorylated after controlled mechanical ventilation. And this is very important, first of all, because it can affect the architecture and can really make the muscle prone to injury. And in fact, we have clear evidence of structural injury, both in animal models of controlled mechanical ventilation. This is electron microscopy, and you have to be an expert to understand that we have here lipid vacuoles, abnormal myofibrils, and that it increases during the days of mechanical ventilation. And now we have human experiments human data showing exactly the same by electron microscopy. This is long-term mechanical ventilation. You can definitely see the injury to the sarcomeres. And in fact, the more you keep a patient on controlled mechanical ventilation, the greater is the percentage of injury in the muscle. And this is important because the more abnormal myofibrils you are having, the less force you can produce, at least in animal models. And I suppose this happens in humans. So to summarize, we have clear evidence that there are at least 3 major mechanisms of injury, like atrophy, oxidative stress, and structural injury. Now, a crucial question is, do partial support modes of mechanical ventilation can protect us from ventilator-induced diaphragmatic dysfunction? And I really don't have the absolute answer. There are no data in humans. What we have in animal experiments, and the first one was by Catherine Sassoon's group, which showed that if we have an animal subjected to controlled mechanical ventilation, take the diaphragm out, and in vitro test the force-generating capacity upon various stimulation frequencies. Here is the force corrected for cross-sectional area. You see that in the control situation, before any just spontaneously breathing animals, you have that force frequency curve. You do controlled mechanical ventilation, total silence, disuse of the diaphragm, and you have a great reduction in the force-generating capacity, and you do assist pressure control, no other information given about the level, the setting, anything, and you get a relative protection, some decline, not very statistically significant, small number of animals, which suggests that activation of the diaphragm, which makes sense, retention of the activity of the diaphragm could be protective. Press support. Well, even here we have more difficult data in animal experiments. We have one study that has only measured proteolysis. And they have been able to show that proteolysis is increased after controlled mechanical ventilation for 18 hours. But if you do press support ventilation for either 6 or 18 hours, it is not increased, the only real finding of that study. Now we have a more recent study by Scott Powers' group, which say again that they used press support, and they have provided evidence that press support could really, depending on how you use it, be either protective or non-really protective. Here you have oxidative stress for hydroxynonenal and calpain activation. So you have upon 12 hours of controlled mechanical ventilation increase in the animals in the oxidative stress. But at the same time, you have the same increase in the press support ventilation. Now, if you go with calpain activation, you don't have a statistically significant increase at 12 hours, where you have at controlled mechanical ventilation. 18 hours don't seem to be very different. But in fact, if you go exactly to see the data, small animal number, I'm not sure that this is not a trend in here. And definitely, if you measure the proteasome activity, you see that at 12 hours, press support was not able to increase the proteasome activity. But at 12 hours, controlled mechanical ventilation was. In fact, it was press control. And at 18 hours, both were able to increase the activity. And of course, the caspase had similar pattern, which suggested that the way they used press support was causing problem, but a little bit later on. It could delay a little bit the problem generated. And in fact, when they measured the force generating capacity, they came out to the same conclusion. Here you have the control situation, 12 and 18 hours of controlled mechanical ventilation, 12 hours of press support, 18 hours of press support. You have a decline, not as much pronounced. So probably it's not the modality per se, but it is how you unload the muscle, because here they have really high levels of press support that they used, and I'm not sure that they were really able to titrate exactly the amount of press support. Now, in bigger animals, we have one study showing that if you use adaptive support ventilation from the very first moment of mechanical ventilation instead of controlled mechanical ventilation, this is adaptive support, this is controlled mechanical ventilation, and this is the transdiaphragmatic pressure developed upon phrenic nerve stimulation, you have really a protection in the force generating capacity. Okay, so when should we suspect clinically ventilator-induced diaphragmatic dysfunction? Clearly, when a patient fails to win, this is when we start thinking about that, and has received controlled mechanical ventilation. Of course, we should really exclude other causes of muscle weakness in the ICU to be sure that this is purely a result of ventilator-induced diaphragmatic dysfunction, like critical illness, polyneuropathy, and myopathy. But to my understanding, ventilator-induced diaphragmatic dysfunction, in fact, might coexist or even aggravate ICU-acquired paresis. You cannot really clearly separate these two. Now, if we are obliged to use controlled mechanical ventilation clinically, we have a TBI patient with traumatic brain injury, and we have to put him on barbiturate coma just to keep the intracranial pressure down and protect him. What can potentially we do? Well, what they have done, they have said that at the animal experiment level, only animals now, that, okay, we have oxidative stress. Oxidative stress is etiologically linked to ventilator-induced diaphragmatic dysfunction. Let's give antioxidants. And they have given vitamin E, an analog Trolox. Unfortunately, this is not allowed to be used in humans. And they were able to restore the force-generating capacity. So if you give it from the beginning and you prevent the development of oxidative stress, you return the reduced contractility back to normal. They have used apocynin, which is a specific NADPH oxidase inhibitor, an enzyme that resides in the membranes and causes oxidative stress. And they were again able, if they give it from the beginning, to protect the diaphragm. Curcumin is a really interesting drug because it is in clinical trials and could be used in humans, and again was protective. And finally, a very specific drug that is in clinical trials, which targets oxidative stress generated by the mitochondria per se, which is called SS31 antioxidant, or Schiller peptide from the investigator that invented it, and it is in fact a small peptide, is really able to reduce the detrimental effects of controlled mechanical ventilation on diaphragmatic contractility in animals. Do we have any evidence that this is true for humans? No real direct evidence. The only very, very indirect evidence that we could get is a study published 10 years ago, a multi-center study in surgical ICUs, showing that the combination of vitamin E and vitamin C could reduce the duration of mechanical ventilation in the general surgical ICU. Is the reduction of ventilator-induced diaphragmatic dysfunction with any beneficial consequences contributing to that beneficial response? I don't know, but this is the only very, very indirect evidence that something is working there. And of course, we have tons of both real data and meta-analysis showing that at least antioxidants are on average safe, so you could try them. So, to conclude, to prevent ventilator-induced diaphragmatic dysfunction, we should use assisted modes of mechanical ventilation as much as possible and try antioxidant supplementation when controlled mechanical ventilation is inevitable. We really don't know what happens when the muscle is not healthy. All these experiments were conducted, at least the animal experiments, in previously very healthy muscles. We don't know what is happening if the muscle is already septic, if we have already critical illness, polyneuropathy, and myopathy. We have a lot of research to do to understand the deleterious effects of mechanical ventilation of the diaphragm. Thank you for your attention.